Okay, hello to all of you. I'm going to discuss the instructions for the Tetrahymena Lab, and this is the last lab of the semester. And this lab is basically about the process of phagocytosis. And we have already learned in the lecture what is the process of phagocytosis. We have studied about endocytosis, and we know that there is three different kinds of endocytosis. There is phagocytosis, when the cells they feed on something solid, pinocytosis, when the cells feed on something which is fluid, and receptor mediated endocytosis. And we have studied the whole process of the receptor mediated endocytosis, when the ligands they first bind to the receptor and how it gets internalized and all those things. So basically in this lab, we are going to study the process of phagocytosis, and there are many eukaryotic organisms which feed by this process of phagocytosis. We are going to study the organism called Tetrahymena. Okay, and these tetrahymenas are basically non-photosynthetic single-celled eukaryotes. Okay, and they take the nutrition by taking solid material by the process of phagocytosis. And in this lab we are basically going to this lab has two parts. First of all, we are going to determine the feeding rate in the tetrahymena. Okay, we are basically not going to use the latex beads, but we are going to use this India ink dye, and this dye is actually going to go and stain the food vacuoles inside the tetrahymena. And how many vacuoles are getting stained by this dye? On the basis of that, you are going to count them, and on the basis of that, we can determine the feeding rate in the tetrahymena. Let me show you some pictures here, like this. If you see this, so this is tetrahymena, and this is stained. This is staining done with the India ink dye, and you can see these are the food vacuoles which are getting stained, and this is what you are going to look under the microscope. We are going to use a phase contrast microscope for this lab, and you will be using bright field, and I will be there in the lab to help you guys to focus everything. So basically, to determine the feeding rate in the tetrahymena, what are you going to do? All the calculation parts, all the statistical part, how to determine if your null hypothesis is going to be accepted or your alternative hypothesis is going to be accepted. All these things I'm going to discuss with you on Tuesday. So Tuesday's lecture will be very important to understand this lab. So in the lab on Monday for section 1, and for the lab on Thursday for section 2, I just want all of you to do the experimental part, and then analysis part we will learn on Tuesday. So in the today's lab, in the first part of the today's lab or this week's lab, you will determine the feeding rate in tetrahymena. In this lab, you will feed them with India ink dye. If the number of vacuoles which are getting stained per tetrahymena increases over time, then the slope of that relationship, beads per minute, is the feeding rate. So I will discuss all these things on Tuesday. So basically, experimentally, what you're going to do. Here are some of the precautions. You can just go over them. Okay. So experimentally, what you're going to do, you are going to take 150 microliters of tetrahymena into a microcentrifuge tube. Okay. And then you are going to add 6 microliters of India ink dye into the same microcentrifuge tube, and you're going to mix the tetrahymena with this dye, cap it and invert it for a few times to mix. So you need to take the tetrahymena and you need to add the dye, and you need to mix them. So what is going to happen? The tetrahymena will start taking this dye inside, and the vacuoles will start getting stained. Okay. Now you have to let this thing happen for 4 more minutes. So basically, in the tube you have tetrahymena and you have the dye, and you have 150 microliters of tetrahymena and 6 microliters of the dye. So this is your tube in which all the dye is going to enter inside the tetrahymenas. After 4 minutes, what you're going to do, you are going to take a slide. Okay, you're not going to do anything to the tube. Let it happen, whatever is happened. You are going to take a slide, and on that slide, you are going to take 30 microliters of Lugol solution or iodine. Okay, 30 microliters of that. And at the 5th minute, so you have tetrahymena with the dye in the tube. Around 5th minute, you are going to, or after 1 more minute, because at 4th minute, you have added the iodine solution on the slide. Okay, after 1 more minute, you are going to take 30 microliters of that mixture of tetrahymena and the dye, 30 microliters of that, okay, and you are going to add that 30 microliter of that mixture on the 30 microliters of the iodine which is there on the slide. Okay, so you're going to take 30 microliters of tetrahymena with the dye and you're going to add it to the 30 microliters of iodine solution which is on the slide. So 1 minute after step 3, 5 minutes after step 2, pipette 30 microliters of tetrahymena bead solution, we are using India ink, onto the slide. Use the pipette tip to thoroughly mix the solutions on the slide. So you are going to mix the these solutions on the slide. Add a cover slip. Thoroughly mixing is essential to kill all the tetrahymena. So at this point, we want all the tetrahymena to die. We don't want them to take more iodine stain inside them. We don't want that, because now we want to determine how many vacuoles they have been stained inside the tetrahymena. So this is your 5th minute point. You have to be very careful because in your tube tetrahymena and the dye is there, okay, and the time period. So basically you have to take you have to count how many vacuoles are going to be stained. You have a time point of 5 minutes, which is this one. Then next time period is 15 minutes. So which means that for 15 minutes you have to see the vacuoles after another 10 minutes. The next third time point is 25 minutes, which means another 10 minutes. Then next time point is 35. So after every 10 minutes you have to after every 10 minutes you have to take that tetrahymena and the India ink dye which is there in the tube. Okay, when you take 30 microliters of that and you add on the slide, when the tetrahymenas come in contact with iodine and you mix them nicely, you kill the tetrahymena. So whatever the feeding has happened, we just want to count that. Okay, I hope you're understanding. I will explain more in the lab. So once you have these tetrahymenas with iodine, you have to first of all look at them. You need to focus the tetrahymena at total magnification of 100x, which means you're going to use the 10x objective. And then once you find the tetrahymena, then you can switch to 40x objective. And it says here dark field, use bright field. Don't get confused. This is from our past experience. Just use bright field. Okay, I will be there to tell you. So you will be first of all using the objective 10x, then you are going to go on 40x. Okay, then inside the tetrahymena you need to count how many vacuoles are getting stained. Okay, and you have to count the vacuoles for at least 8 tetrahymenas. Count the beads in 8 tetrahymena. Okay, so this is what you have to do, and after that you have to discard the slide in the slide drop, and then you have to take the readings at 15 minute, 25 minute and 35 minute. Okay, and the data analysis graphing we will discuss on Tuesday. If you see here keep going, here is a table. Okay, all your results you have to write down here. So you are going to count the stained vacuoles in tetrahymena after 5 minutes, after 15 minutes, 25 minutes and 35 minutes. This is your first lab. This is the first part of the lab. In the second part, what you have to do, basically we are going to see the effect of, in the first part we have determined the feeding rate of tetrahymena. That's what we're trying to do. In the second part, we are trying to see the effect of different factors, which are basically the independent variables in the experiment. We are trying to see the effect of different factors like temperature, effect of protosol, effect of ethyl alcohol, effect of caffeine. We are trying to see the effect of these factors on the feeding rate of tetrahymena. So in this experiment, so every group is just going to use one factor. You can choose either temperature, you can use protoslow, you can use ethyl alcohol, you can use caffeine. Only one factor you have to choose, and then you are going to determine what is the effect of this factor on the feeding rate of tetrahymena. So in this experiment, the independent variable will be, if you have chosen temperature, then temperature is the independent variable. If you have chosen protoslow, then it is the protoslow, and your dependent variable is the feeding rate of tetrahymena. So basically, if you have chosen temperature, you're trying to see what is the effect of temperature on the feeding rate of tetrahymena. Protoslow is basically slows the swimming of the tetrahymena. You're just trying to see what is its effect on feeding. Ethyl alcohol, what is the effect of ethyl alcohol on the feeding rate of tetrahymenas? Caffeine basically can cause smooth ER and mitochondria to release calcium and another secondary messenger called cyclic AMP. So you are trying to see how does that affect the feeding rate of tetrahymena. So how to do this experiment? I'm going to discuss that. Okay, so if you have chosen anything out of these, if suppose you are trying to see the effect of ethanol, caffeine, protoslow, or high potassium, so what are you going to do? So at a treatment station, you will take two tubes and you're going to label them A and B. Add 100 microliters of control medium in tube A and 100 microliters of treatment solution to tube B. So the controls will be given to you. So if you have chosen caffeine, then in the tube A, you are going to add control medium, and in the tube B, you are going to add the caffeine. And then to each tube, you are going to add 100 microliters of tetrahymena. After this, everything is same as the first part. You're just introducing a factor and you're trying to see how it affects the feeding rate. After that, everything exactly is same. And then you are going to add to both the tubes, you're going to add 6 or 8, both are fine, 6 or 8 microliters of the India ink dye to both the tubes. Then you are going to do the same thing. You are going to cap it, invert it, you're going to basically mix them. And here starts your 0 minute. And then you have to do the same thing, basically after same thing as you did in the first part. After 4 minutes, you are going to take a slide. You're going to add 30 microliters of iodine. And when the 5 minutes are complete for your tube, then you're going to take 30 microliters tetrahymena. You're going to mix them on the slide, put the cover slip, and you are going to first of all focus at 10x. Once you focus it, you are going to then change your objective to 40x. You are going to look inside the tetrahymena and you're going to see how many vacuoles are getting stained with the India ink dye. That's what you have to do. And you have to do the same thing for the different time periods like 5, 15, 25, and 35. Exactly same. And suppose if you have chosen the temperature, then what you're going to do at the treatment station, label 2 tubes A and B. So add 150 microliters of tetrahymena to each tube, and so basically you have 2 tubes with 150 microliters of tetrahymena. Now it is up to you what kind of temperatures you have chosen. Maybe you can choose something like ice. You want to see the effect of ice and room temperature, then you can keep 1 tube in the ice, 1 tube can be on the room temperature, and then again I mean how much, like around 10 minutes you can keep your for several minutes. So place your tubes in their temperature. It all depends what kind of temperature you have chosen. I would say 10 minutes is good enough. Place your tubes in their temperature regimes for several minutes, then add. So basically you're going to expose the tetrahymena to that temperature, whatever the temperature chosen, and then add these India ink dye, mix them, and you will be doing again the same thing. Like after 4 minutes, once you add these India ink, it's a 0 minute time period starts. After 4 minutes, you basically take your slide, add the iodine, and then take 30 microliters of tetrahymena, mix them, and put the cover slip and watch them under the microscope. Exactly same. And here is your table where you have to write down your data. Okay, so this is what it is. So whatever the treatment you have chosen, you have to write down treatment A, treatment B. Suppose you have chosen caffeine, then treatment A is control, treatment B is your caffeine. If you have chosen temperature, now if maybe temperature A is ice and treatment B is your room temperature, and this is 5 minutes, 15 minutes, 25 and 35 minutes, And here you are going to write down all the values. You are going to write down how many vacuoles you have found. You have to count the vacuoles in at least 8 tetrahymena. So you're going to write down the data here. That's all I want you to do in the lab, and the rest part I'm going to explain on Tuesday. And I will be there in the lab to help you.